dry heathlands in the Netherlands. Stunning landscapes providing habitat for a wide variety of rare species. Species of birds, plants, mammals, butterflies and reptiles. Unique and diverse. However, many of these species have become endangered as a result of habitat fragmentation and reduced habitat quality. The manifold of dynamic processes formerly present in the large interconnected areas of cultural heathlands, formed as a result of sheep grazing, are increasingly reduced or lost. More highways, intensive farming and urbanization decrease habitat suitable for species characteristic to heathlands and cause enrichment of soils via atmospheric nitrogen deposition. Since it's virtually impossible to solve these problems in short notice, nature managers are increasingly implementing grazing management strategies to enhance landscape heterogeneity and therewithal maintain biological diversity. The role of large herbivores is to control succession by grazing the encroachment of dominant, fast-growing grass and shrub species. Herewith, an open mosaic landscape will be formed to maintain biodiversity levels as desired. Oftentimes, floristic diversity is measured as indicator for nature quality in heathlands, but side effects of grazing management on microfauna is frequently lacking. This episode will highlight the effects of grazing management on dung beetle communities in Dutch heathlands. The dung beetle. Oftentimes, this animal is considered to be a dirty, feces-creeping creature. Yet, this insect contributes to many ecosystem processes. While herbivores control forest succession and grass domination, accompanying dung beetles play a major role in relocating organic material. Moreover, they accelerate soil mineralization rates, secondary seed dispersal, all doing this by relocating feces parts. Since grazing management is increasingly adopted as nature conservation strategy, higher quantities of feces are likely to be released to the ecosystem. This implies favorable circumstances for dung beetles to colonize these nature areas. Depending on the grazed species, different types of feces will enter the environment. To this end, we formulated the following research question. What is the response of dung beetle communities in heathlands to different feces types? Our first hypothesis is that there is a difference in preference by dung beetles for feces of specific herbivore species. Our second hypothesis is that preference for feces by dung beetles is determined by the difference in feces quality of different herbivores. In order to predict the differences in feces quality and differences in feces preference by dung beetles, we carried out a literature study. According to Hensky and Kaempfer, moisture content, feces structure and nutrient content are main factors driving feces quality for dung beetles. Schultz confirms part of this theory. He mentions that moisture content increases feces quality. Dung beetle mandibles are not fitted for chewing and therefore not suitable to consume solid material. Furthermore, Finn and Giller mention that different herbivores have different digestive properties, influencing the fiber content. Herewith, increased fiber content is considered equivalent to decreased feces quality. Moreover, Hensky and Kaempfer keynote that nutrient content determines nutritional value, increasing feces quality. The overall feces quality is expected to determine the beetle abundance. Based on these findings, we predicted the following differences in feces preference by dung beetles. Dung beetle abundance is most likely to be higher on cow and sheep feces, since these herbivores are ruminants. Their feces is expected to have higher nutrient availability and finer texture compared to horse feces. As cow feces is assumed to be very moist, dung beetle abundance is expected to be highest for this group. Also, we predict higher dung beetle diversity at cow and sheep feces compared to horse feces. We think so since feces of these ruminants are expected to contain higher edible fractions, facilitating multiple species of dung beetles. Our third and fourth prediction refer to feces manipulation. When feces becomes saturated with moist, we expect sheep feces to attract more dung beetles due to increased availability of edible fraction. When fibers are added, we expect that beetles will be most attracted to cow, then sheep and last horse feces, since nutrient levels of these feces remain higher despite coarse structure. In order to test our hypotheses and predictions, a correlative field study was carried out in combination with a manipulative lab experiment. We will now demonstrate the design and results of the correlative study. The correlative field study was performed at the Ginkelse Heide near Ede. This choice was based on the presence of dung beetles and 3 herbivore species observed during the pilot study. The area proved to be a proper site to test if dung beetles have an appetite preference for dung of specific herbivores. Although bovines are absent within the area, these grazes are present in neighboring areas that fall within the beetle dispersal range. To determine dung beetle feces preference, pitfalls filled with dung were placed to capture them. The following setup of the pitfalls has been applied to assure that beetles had free choice of dung. This means that within one block all varieties of dung will be present, whereby each feces type is placed in 2 pitfalls per block. 3 types of dung were randomly placed in 6 pitfalls and 2 empty cups were included as control. Samples were stratified throughout different vegetation types: heather, bare sand and grass. Per strata, 5 blocks were randomly set out and replicated once in time. This resulted in 240 pitfalls to be analyzed in total. The distance between the pitfalls within each block was chosen to be at least 2 meters to prevent pitfalls from influencing each other. The blocks were situated at least 30 meters from each other. After 24 hours, the first 120 pitfalls were numbered and extracted from the field to be transported and analyzed in the lab. The second round of pitfalls were brought into the field, while the first pitfalls were extracted and again collected the next day. Pitfalls were examined in the lab by counting the captured number of dung beetles per species. This step in the experiment was quite dirty, since pitfalls were filled with feces. For this reason, remaining dung dishes were taken care of as soon as possible by a professional dishwasher. During the analysis, data on beetles per pitfall were saved as Excel file in the computer, but in an SPSS adaptable format. In total, 445 beetles were found throughout the 240 pitfalls. To analyze our data, a generalized linear model was applied using a chi-square test. This was chosen since we are dealing with count data, thus a Poisson distribution. Differences between groups are tested with a Cheffe post hoc test since the data is unequally distributed. The generalized linear model showed significant effects of both vegetation type and feces type. Also, the interaction between the two appeared to be significant. Sampling day and block were included as random factors in the analysis. Of these two, only sampling day had a significant effect. Therefore, block is not included in the further analysis. Post hoc testing with the Cheffe test resulted in the following graph. As the data are not normally distributed, it was chosen to visualize our results by means of box plots. Differences between vegetation types, indicated with capitals, show a significantly lower beetle abundance in sand areas. This could be explained by the lack of cover, which protects the beetles against predators. Within each vegetation type, it turns out that the qualitative pattern is the same. Dung beetle abundance is significantly higher on cow and sheep feces, compared to horse feces and control. However, abundance on sheep feces seems to be higher in each vegetation type. The p-value of the difference between cow and sheep is 0.09, which indicates that with a larger sample size, this difference may actually turn out significant. This can be explained because both cow and sheep are ruminants, which are expected to contain higher edible fractions in their feces. Moreover, specialization of sheep feces by dung beetle may be present in this area, as sheep are the dominant grazers within the area. Preference for horse feces is basically absent, which can be explained by the high fiber content, which means that it contains a low edible fraction. 4 different species of dung beetle were distinguished in this research. Overall, the number of species showed the same pattern as species abundance, with a higher number of species on sheep and cow feces, and lower numbers on horse and control pitfalls. However, it is possible that this pattern is caused by difference in abundance, as it is more likely to find more species when more beetles are caught. By means of assimilation in estimate S, we can see that with an increasing sample size on all 3 feces types, the 4 beetle species are found. Based on the error bars, it seems that there is no difference between the 3 feces types. Only the control differs, but here very few dung beetles were found. To determine the differences in feces quality, several properties of the feces had to be measured. By drying the feces samples of bovine, horse and sheep at 105 degrees Celsius, moisture content was determined. The samples were weighted before and after drying to calculate the percentage of moisture. After drying the samples even more at 550 degrees Celsius, they were weighed again to measure the organic matter content in percentages. For all 3 feces types, nitrogen and phosphor percentages were measured by means of a chemical analysis to determine the nutrient content. Edible fraction is determined by sifting the feces of the different herbivores. For this, 5 different sizes were used, with a 0.05 mm as smallest sift size. Based on literature, we assume that particles smaller than 0.05 are edible for dung beetles. It should be noted that all feces properties are measured as mass percentages. Few significant differences in feces quality were found. As organic matter and nitrogen were normally distributed, an ANOVA with a Tukey post hoc test was used to analyze these data. For the not normally distributed data of phosphorus and moisture content, a Kruskal-Wallis test with a Scafé post hoc is used. Moisture content appeared not to be different among the different feces types. Also no difference was found for nitrogen content. However, for nitrogen content a qualitative pattern can be seen, in which sheep has the highest nitrogen content, followed by horse and lastly cow. The same qualitative pattern can be seen for phosphorus as organic matter, but here there are some significant differences. All feces types had a significant different organic matter, and sheep had a higher phosphorus content than cow and horse. Fiber content also showed the same qualitative pattern. However, here it should be taken into account that cow feces contained quite some stones and sand, which influences the weight quite strongly. For this reason, the mass percentage of the edible fraction might not be accurately estimated by the sifting method. Overall, the sample sizes used for these analyses were limited, due to the tight time frame and extensive labor needed for the sifting. Increased sample sizes might show more significant differences in feces properties.
But what about the power?
The 4 samples that were analyzed seems few to determine characteristics of different feces. Hence, a power analysis was done to determine what sample size would be necessary to find significant differences. Since the data was not normally distributed, a regular power analysis was not possible. Instead, the sample size was enlarged in a simulation to determine when the probability level for all characteristics would be under the threshold value of 0.05. The black line represents the difference with the largest variance between the feces types. Duplicating the samples revealed that all characteristics would be significantly different with a sample size of 12. To uncover causal relationships, a manipulative study was conducted as well. We will now demonstrate the design for this. From literature, it became clear that feces preference by dung beetles may depend on moisture content, texture, and nutrient content of the feces. To test for the effects of these characteristics within the lab, we did 3 separate cafetaria experiments, one under normal conditions, one with moisture saturation, and one with texture saturation. This results in 3 one-factor designs with 3 levels each, respectively cow, horse, and sheep feces. The manipulations are tested in 3 arenas. Each of them consisted of 3 compartments and a mid-compartment with 3 openings. This mid-compartment was chosen for to reduce the effect of random marching by beetles. By this, we consider movement to a certain compartment as actual preference for that feces by the dung beetle. In each experiment, 120 beetle have been tested by releasing groups of 5 randomly selected beetles in the center of the arena. Beetles are left without food for a day before testing, to trigger urge for dung. After insertion to the arena, beetles are given 5 minutes to choose between the different feces types. To reduce carryover effects, beetles are used only once and rested for a day between each experiment. The data retrieved from the manipulative study was analyzed with a generalized linear model. For all treatments, feces type had a significant effect. Again, differences between feces types were analyzed with a Scafé post-hoc test. Feces in natural conditions showed the same pattern as in the field, with highest beetle counts on the sheep, followed by cow, and with the lowest counts on horse. After applying moisture saturation, this pattern changes. In this situation, horse feces attracts more beetles than cow feces. An explanation for this is the increased food availability for beetles when edible particles are dissolved in a solution. As cow feces is more liquid than horse feces, edible particles are more likely to be already in a solution and available for the beetles. However, horse feces is much less liquid, and therefore moisture saturation can increase the food availability. Saturation with fibers to change the texture also changes the main pattern. Again, horse feces attracts more beetles than cow feces in this case. This can be explained by the higher nutrient content of horse and sheep feces compared to cow feces. When texture saturation makes feeding for the beetles more difficult, food with higher nutrient content might be more attractive. Based on the results of our correlative field study and manipulative lab experiment, we concluded the following. Sheep feces attracted more dung beetles than was hypothesized in advance. However, this can still be explained by the mechanisms thought of. Sheep feces contained a higher nutrient content and edible fraction than expected, making it more attractive. Both moisture content and texture appear to be driving factors determining feces quality. We speculate that these characteristics differed among feces types. Although this cannot be confirmed due to the low sample size, observed patterns of these feces analysis suggest these differences do exist. We assume that the beetles aim to find food of high quality, which is easy to consume. Moisture content contributes to the consumption ease, whereas nutrients contribute to the nutritional value. These characteristics, together with the edible fraction, determine the food availability. These outcomes imply that sheep grazing management supports dung beetle communities better than grazing management with horses or cows. If adopted, sheep grazing management indirectly contributes to processes such as soil mineralization and seed dispersal, which subsequently benefits the ecosystem as a whole.